Hi, you guys, and welcome to Chapter 15, Circulatory Physiology, and this is Part 1. We will stretch this over 2 weeks. So this first picture here is an open system and a closed system of circulation in different organisms and different animals. The one on the right, it would be us, and this would be an insect, for example, a different kind of animal that has some sort of pump. And it collects fluid here, from the abdomen, and it'll bring it up into this pump-like and then pump it back out again. So both sides can sort of pump out and collect up fluid and recirculate that fluid. That's an open system. We have a closed circulatory system where the blood is entirely enclosed in vessels that are going to connect to each other and then go back to the pump. Go back to the heart. So it's interesting to see how those would work. In this system that we have about 5 liters worth, and those are the ones that are circulating, and we can distribute them in different ways. This diagram I like a lot. This words are cut off a little bit. The circulatory system in parallel. When you draw it like this, you get a really good idea of how we could regulate that flow going to different organs. So if you drew it so that you had the right heart in blue because you have deoxygenated blood and it's coming over to the capillaries of the lungs and it's loading up, and so it's leaving those capillaries red. And it's coming to the left side of the heart now and pumping to the rest of the body. And this would be the aorta here, because that's the biggest vessel leaving the heart, and you'd pump it and it would go to all these different organs and it branches. Now, if you decided to alter the radius of this one and kind of close it down, what would happen is that less blood would flow here and more blood would flow into this other one. And if some of it backed up, it would flow into the other one. So you still have a closed system that blood has to go somewhere, but we can regulate. So we can regulate blood going to the digestive system when you're out running a marathon, and you can shunt it over to the digestive system when you're eating. So you have that possibility of directing blood to the location it needs to be. And vessels are going to be able to do that. They're going to be able to narrow up, and that term is vasoconstrict, or they're able to vessel dilate. And if they vasodilate, they're going to allow more blood to that particular organ. That's why I think this diagram is really good. Continuing with blood vessels, we have the arteries that are going to deliver blood to the tissues. They're delivering oxygenated blood to the tissues. Capillaries are where the exchange is going to happen. So in this diagram, we have an artery, 2 arteries arriving at a capillary bed, the site of exchange. So nowhere else. This here is transport. This here is transport. It is only when you get to the capillary, we call it a capillary bed, that exchange is going to happen, meaning I'm going to be able to deliver some of that oxygen and pick up some of that carbon dioxide. The veins then, once it's gone through this capillary bed, the veins will be able to pick up that blood now that's devoid of that oxygen and then again transport. So this is transport again. There's no exchange happening here. Good. If you were to look at it on a real tissue slide, a histology slide, how would you recognize which the artery was and which the vein was? So you would know that the one that holds its shape, the one that has this, thicker musculature layer in this wall here is an artery. And the one that has a thinner layer here to here, this thin layer here is floppier. It's going to sort of collapse on itself when you prepare a slide like this. So the floppier one is the vein, less muscle, and artery has more muscle and it's going to hold its shape. There's some common characteristics to all blood vessels, and we want to look at that. You know, are they all the same? The vein is, how is it different from the artery that's different from the capillary? They all have an inner lining, and that inner lining, endo, inside lining, the thelium, they all have an endothelium. So it is a different word for an epithelium, which is a liner, but it is the innermost lining of that. Think of it as a hose. If you have a hose like this, and the inside lining of that hose here, innermost lining, would be the endothelium. They're important because they have other functions other than just holding that blood. And they can secrete paracrines, they can secrete hormones, and in doing so, they can regulate blood pressure. They can vasoconstrict or vasodilate. So let's make one dilated here. You'd have this larger opening here. So endothelium, very important. There's a picture, a better picture of vasoconstriction and vasodilation, in which you're moving greater volumes or lesser volumes of blood towards organs. So we had the endothelium. Next, we're going to have smooth muscle. Smooth muscle is the muscle that allows for the vasoconstriction. So the endothelium is secreting that hormone, that paracrine, that can cause the vasoconstriction, but the actual smooth muscle is the one that is causing that narrowing. It's going to increase resistance, it's going to increase pressure. Capillaries don't have smooth muscle. They're not able to do that. They only have one function, and that is to allow substances to go in and out. So you wouldn't want to put a muscle layer on there. This diagram seems to have a lot of information on it, but I like it a lot, and I'd like you to look at it. It is showing the differences in vessel size and thickness. So the numbers I'm not concerned with, the mean wall thickness, just kind of if we look at how these different tissues are distributed in each one of these, it would be helpful. So we want to look at an artery and a little artery, an arterial, a capillary, a vein here, and a little vein, venule, and how those are going to look. So in an artery, notice how it seems to have a thick wall here. So it has an endothelium. We said all of them have an endothelium. They have elastic tissue. And why do they need elastic tissue? Because it'll stretch if it vasodilates, and then it has to be able to retract back again. So it has to have that elasticity. And only large arteries and large veins will have that. Notice that little arteries, arterioles, venules, and capillaries do not have that elastic tissue. But do they have muscle? Do they have smooth muscle? Well, the arteriole does a little bit. It loses it in the capillary, none in the venule, and some in the vein. So the diagrams help a lot. So a vein is similar to the artery, but much less smooth muscle, much less elastic tissue. The arterioles are responsible for vasoconstriction, so you would want smooth muscle in there. And they're under the effect of a hormone or a paracrine, so they don't actually need the elasticity because they will be able to control the diameter of that vessel. Okay, so it takes a little staring at this for a while to get a sense of how these differ. A little bit more on large arteries. Large arteries are elastic. So think of something big like the aorta. As soon as blood hits that aorta, it means that the ventricle contracted. And if the ventricle contracted, it just shot out at high pressure quite a bit of blood into that vessel that's not rigid. If it's not rigid, it's going to actually stretch. So this diagram is showing a ventricle that used to be this big is now going to contract. If it's going to contract, it's going to have pressure pushing out into it, and this vessel that used to be, this wide has now expanded. And so we think of large arteries being elastic, and the next step is this one down below when the ventricle now is going to relax. It's going to fill again. And this vessel that used to be this size is now, through its elastic tissue, going to retract. So what does that do? Think of it as a balloon that you expand and then you let relax again. It works kind of like a pump. So we say that these large arteries, yes, they're elastic, but they also aid in the moving of blood. They act like a mini pump. Very important. They act like a pump. Smaller arteries don't act like a pump. They are more resistance vessels. If you think of how much branching goes on into these smaller arterioles, it's an intense branching, and it creates resistance because you have blood now branching out into lots of different vessels, and you're going to need force to be able to push the blood through that. You're going to need pressure to get it through all those vessels. Arterioles will be able to determine the distribution, as I said. They control blood flow to capillary beds also. So they will control it and send blood to where it is needed at that particular time. Going back to total peripheral resistance, TPR, this is a diagram that gives a better idea of that. So if this was a larger artery, here it's branching out and branching and branching and branching. Can you see how just the friction against the walls of all these arteries is going to cause resistance? So your total peripheral resistance is that resistance to flow given the intense branching. It is also used, think about when an individual has atherosclerosis, and these vessels, let's say, have accumulation of cholesterol and fat in here, and now you've further narrowed these vessels. And if you further narrow those vessels, then the total peripheral resistance is going to go up. And if the total peripheral resistance goes up, then you're going to need more pressure in order for that blood to flow past this area. So more pressure means your heart has to beat harder. It's going to have to contract more forcefully to be able to overcome the total peripheral resistance. Back to arterioles, they change the diameter. And so we go back to my little drawing where you remembered where I could narrow it here. If I narrow it and open here, I'm going to be distributing to different organs. They're not drawn in here, but I'm actually distributing to a capillary bed of one particular organ. Maybe I've opened this up here also, and I have another organ here that is getting irrigated by this particular capillary bed. So arterioles are the ones that can change that diameter and control that blood flow. Capillaries. Capillaries are exchange vessels. So I've said that before. This is where you're going to distribute oxygen, pick up CO2, and pick up waste and distribute other ions that are necessary in that particular location. They're also the connection between the arteries and the veins, right? It's a closed system, we said in the beginning. So they are what connects those two, the venous and the arterial blood. They are tiny. They are about 0.5 micrometers. This is in diameter. So if you think of a hair is about 100 micrometers. So it's about half the size of a hair. This is a really nice photograph of them. They're really tiny. They're so small that if you took a cross section of a capillary like this, you would see that a red blood cell would barely fit. You can't have two side by side here because it wouldn't fit. So that's how narrow a capillary is. You have a single cell red blood cells traveling like in a train fashion, one after the other, follow the leader, only one at a time. So I always ask with students this, is every cell of the body close to a capillary, you know? I mean, think of how many cells you have in your body, and you would need an awful lot of capillaries to be able to serve, so to speak, every cell of the body. So Is every cell of the body close to a capillary bed so that it can get oxygen? And the answer is yes. No cell of the body is farther than 4/1000th of an inch from a capillary. That's 0.01 centimeters. So yeah, if you want to be a cell that receives oxygen, you've got to be near a capillary bed. Therefore, capillary beds are immensely abundant in your body because they have to feed every single cell. It turns out that if you were to iron it all out, all the capillary beds in your body, it's about 600 square meters of surface area. Like, I think that's like a tennis court. And it's a huge surface area for exchange, and that's what you need. You need to be always serving those cells, so that surface area is amazing. The other interesting thing about capillary beds is that you can control the flow. Let's say there is no need, you're sitting down, there is no need for a lot of blood coming to muscle of your legs while you're sitting. You're not running, you're not doing anything. So maybe you could kind of bypass that capillary bed and not have to irrigate every single region. So you could do something like this, and you could arrive at a small arteriole and then find your way to a venule and move on without really having to fill all this capillary bed, which is the example given here. So you can control the flow, and they don't have muscle of their own to vasoconstrict. So instead you have little valves, right, like a sphincter. So if I close a sphincter here, that's a little bit of muscle that I can shut. If I have a little bit of muscle here, a little bit of muscle here, maybe here, right? All these are sphincters, and I close those off, and I can bypass all this other region and avoid irrigating a region that really has, at that time, no need. Written out for you, open precapillary sphincters allow blood into capillary beds. So these are the precapillary sphincters not allowing blood into the capillary. And closed precapillary sphincters shunt blood to other capillary beds. So by not irrigating this blood will be able to go somewhere else and irrigate a bed that needs it at that time. The meta-arterial allows blood flow to pass. So the one region that never closes, because you have to connect the artery to the vein or the arterial to the venule, that one freeway needs to stay open. That is the meta-arterial. The one that has to stay open regardless if you're going to work with a closed system that connects arterial blood to venous blood. There are different types of capillaries depending on how leaky you want them. And notice that there are videos on this, and these videos have been posted in the other section of Canvas, so you should look that up. This capillary here is a continuous type of capillary, and it has an endothelium, the inner lining, and that's all it has. And this is a cell, right? And this cell is very close together to the other cell here, and that's what makes up this entire tube, right? Cells. You can actually see the nucleus of some of them. So they sit very close to each other, and they form this tube, which is a capillary. It's continuous because this connection here is pretty closed, right? It doesn't look a whole lot leaky. It does leak a little bit, but not a whole lot. So we want to compare that to the other types of capillaries. A fenestrated capillary has the basement membrane, the lower part of these actual cells. We peeled away the top part of the cell. The lower part of the cell has all these holes, right? And it's very porous. So it's going to allow fluid to leak through. And these are called fenestrated capillaries. They have large pores, and depending on what type of capillary and what region of the body they're in. Sometimes you have to filter a lot. If you have to filter a lot of fluid, the kidney's filtering a lot of fluid, you're going to want something that is particularly leaky because you're draining fluid that gets filtered and then bringing it back into the body again. Look at those videos. The third type is a sinusoidal capillary, and this one is a very, very leaky capillary. This one's found in the bone marrow. Think of bone marrow, how you make cells and the cells migrate out of the bone marrow. So this has got to be very leaky in the liver and in the spleen. So they have these holes in them. They're 5 times wider than a continuous capillary. Plasma proteins need to cross these. Gosh, we've always talked about proteins staying inside a cell and they're negative and they stay inside the cell and they can't get out. Well, in this case here, through this kind of cell membrane, they can get out. So this is sort of the exception in sinusoidal capillaries. And a video for you to watch. Think of how once blood gets to a capillary, it's going to be going quite slowly. And that's going to allow time for exchange between the blood and the interstitial fluid, right? If you've taken this big hose and then you've branched it into a narrow one and then branched it into a narrow one and then branched it into a narrow one and you go narrower and narrower, it's pressure is dropping and velocity is dropping. So it's slowing down. And it's a good thing for it to slow down because if it whooshes past that capillary bed, there's not going to be enough time for that oxygen to leak out into the interstitial fluid and to pick up that CO2. When arterial flows into many capillaries, it's going to slow down as it enters the capillary bed. So let's look at it in a graph. This would be the velocity of the blood flow in centimeters per second. How many centimeters it's flowing per second? And you'd want to look at that in the order. Arteries, little arteries. Capillaries, little veins, and then the vena cava. And you know that in the order, it's going, around 30 centimeters per second. And as it starts to get narrower and smaller, the velocity is going to drop. And it's going to be lowest here in the capillaries when exchange has to happen. And then you start to group little veins into bigger veins. They join up together again, and the velocity will go back up again. That velocity is about 0.03 centimeters per second. Very slow, as opposed to the 30 that we started with. Capillary exchange takes place how? Between the endothelial cells, it's called paracellular pathway, or through the endothelial cells, that's endothelial transport. So do you understand the difference? Let's go back one. So these are the cells that make up that capillary. It's either going to come between the cell and be able to ooze out there, or right through the endothelial cell. So let's look at those. Through the membrane, this is coming in. This is an IgA. This is an immune molecule that is coming up against the membrane of this epithelium of the cell. It's pushing its way, blasting its way through into the cell. Now this is inside the cell and it goes out the other end. So this is how it's moving. So it's going right through the membrane, not limited to a leak that's between two cells. Smaller dissolved solids and gases can also move by diffusion through or between the cells, both ways. This depends on their size. So if it's very tiny, it's going to be able to do it very easily. And if it's lipid soluble, it is easy to do because it is a fatty lipid layer. harder if it's water soluble. Filtration and absorption. Fluids can leave a vessel by bulk flow, resulting from hydraulic or osmotic pressure, and I'm going to show you how that works. So in the terminology, when we're moving out of the capillary, think of it as a soaker hose. If you're a gardener, you have a hose in the garden that's got little holes in it, and when you put the faucet on, water shoots out from the little holes and it irrigates your plants. So fluids can leave by bulk flow, and it results from the pressure. The higher the pressure, the more the flow. And when it's leaving the capillary, we call it filtration. So anything leaving is filtering. If it's the other way, those little holes are receiving fluid that's coming in, it's absorption. So coming in is absorption and leaving is filtration. So the next question is, well, which capillaries absorb and which are the ones that filter, correct? Most capillaries do both. The net filtration is at the arterial end, and the net absorption is at the venous end. That movement is going to be, let me go forward a little bit. I think what I want to do is actually stop right here. So let's stop at bulk flow, and I'll do a separate video for that. Thanks for listening.